большущее, Дима. Спасибо большое. Я жду студентов. Да, давайте. До свидания. Спасибо. Хорошего дня. Так, так. Пока никого. Guys, where are you? I am going to start a lecture. So this is in a regular time and in a regular room number 3. Good morning, everyone. Так, ты одному читал студенту, да, Юра? У меня пока никого. Okay, guys, I'm not going to wait until you come because maybe you will listen to this lecture later on. So I should have enough time to cover the basic topics of our lecture. So today we are going to discuss antimicrobial drugs, but particularly we'll pay our attention on antibacterial. So Suraj Maurya, I would like you to tell me whether you hear me very well or not, because I have some troubles with the OBS studio program. Confirm that you hear me very well, okay? So then we may have helminths and as a infectious agents, and the drugs that affect them are called anti-helminthic drugs. And finally, we may have acellular organisms that are viruses. So, and the drugs that affect viruses are called antivirals, briefly. So here you can see a table with that type of classification, where the drugs that we use for chemotherapy are classified according to the agents. And you can see here, and some of them we have touched or we have studied a bit already. So we have touched some antifungal drugs, and you can see here that some most common groups of antifungal agents or antimycotics are shown, and that affect the different fungi. Then the second group that you are familiar with already is antivirals or antiviral drugs. That's what we are studying right now. And you have to know that the way how they are developed is quite different from others. Why? Because antiviral viruses are intracellular parasites, so we have to mainly affect them when they are already inside the human cell or at the very first stages of the interaction between viruses and the cell these are going to infect. So then they are quite different. And this week we studied them and we discussed the targets for antiviral agents. So and now we come up to the drugs that may affect bacteria. And you can see here that we have a lot of different types of groups that here we can see classification of them regarding the targets they reach in the bacterial cell. So it is following. So today we'll concentrate, as I said, on the antibacterial agents. Within those agents, we may distinguish some groups. And of course, the very first one is antibiotics, the most common. Uh, uh, and these are the naturally occurring microbial products. So then we may have a chemotherapy group of antibacterial agents that are called chemotherapeutic drugs. They act against bacteria, but they uh, are different from antibiotics. They are products of chemical synthesis with diverse structures and activities, and those chemical compositions never occurred in nature. And here we also may mention and a sort of antibacterial agents. They are bacteriophages. These are the other type of organism that may kill bacteria by infecting them. So in antibacterial agents, my dears, come back to this. We give short description of chemotherapeutic drugs and bacteriophages and some others, and then the rest of the lecture will be devoted to antibiotics. So what we uh, have under antibacterial agents that are not antibiotics. So here we may have antiseptics. These are the chemicals applied to the skin, wound, and mucous membranes, and they are not antibiotics. And the important thing that they don't act specifically on the bacterial cells, but they can uh, inactivate or damage any types of cells, including human cells. And also in this group, we have disinfectants, the chemical agents that are usually used to inactivate or destroy microorganisms on inert surfaces, so desks, walls, and so on, floor, and some instruments, and so on. Then also in this group, we have a group of probiotics. These are live microbial organisms, beneficially affect the intestinal microbial balance, but generally not only intestinal. So prebiotics are non-digestible substrates, which improves the growth of bacteria in the colon. So usually probiotics are given together with prebiotics or uh, in order to improve the growth of bacteria that we send to the, for example, GIT. So bacteriophages are uh, living organisms, usually of particular nature. They're supposed to be lytic bacteriophages. So why lytic? Because they need to infect bacteria, as far as you see here. Then they supposed to degenerate the bacterial cell, or multiply there, and when they release from the host bacterial cell, they will rupture it, so the cell gonna die. So that's why those bacteriophages can be used as therapeutic drugs against intestinal enterobacterial infections, or quite, you can see it as the bacteriophage. So, example of it. Or quite often, they are very effective against chronic uh, staphylococcal infection. They also can be used as chemotherapy, some therapeutic drugs. Few words about probiotics. These are live, non-pathogenic bacteria. So, but they are good bacteria that are very helpful. So, they may help in recolonization of the particular sites of the body if they are colonized but by facultative pathogenic bacteria, those that may live in harmony with us, but from time to time, under some condition, they may cause diseases. We would like uh, them not to overgrow in the colon. So that's why when we use probiotics, we think that they decrease the or inhibit the growth of facultative pathogenic bacteria. So it really they return the normal flora after, for example, antibiotic therapy uh, due to some bacterial infection. Then they may be effective in enhancing colonization resistance to pathogenic bacteria. And also they are effective in excluding the pathogens from the binding sites because they compete with the uh, highly pathogenic or facultative, not so highly pathogenic bacteria in the normal flora, in the sites where normal flora occurs, and they have very high possibility because they are sort of a result of genetic engineering. They have very high capability to colonize the gut, and they also may produce some substances that inhibit the growth of pathogenic bacteria. So they compete with facultative pathogens and pathogens for food ecological niche that they get. And of course, they may help in reducing the inflammatory response against pathogens. So that's why probiotics, that is quite important part of the supporting therapy, especially when we use antibiotics. So here you can see the examples of probiotics. For example, Biosporin. This is a combination of Bacillus subtilis and Bacillus ligeniformis. Uh, then another example as Acilact. So it consists of three different strains of Lactobacillus acidophilus. And also we have a mm, and as an example, we have so-called colibacterin. So this is a drug or this is a preparation that consists of uh, very helpful non-pathogenic E. coli. But also we may have probiotics that are combined, made up of bacilli and E. coli, for example, or they mm, are the combination of several different types of bacteria. So that's the short description of other approaches that are used in antibacterial therapy. But from now we will discuss more antibiotics because that's how commonly we call all antibacterial drugs. What are antibiotics? So as you see here, the definition to antibiotics had been developed very far ago, around 1940. by uh, Waxman. But then, not so much really changed since that time in definition and understanding what are antibiotics. Antibiotic is a chemical substance produced by microorganisms which has the capacity to inhibit the growth of bacteria or even destroy bacteria and other microorganisms in dilute solutions. My dears, I would like to underline in dilute solutions. It means the concentration of this substance should not or may not be very high, because you understand that theoretically we may get any substance that in very high concentration will kill bacteria. But then in that high concentration, this substance may also heavily affect the human cells or host cells or host metabolism. So then it won't, this side effect will not allow us to use this substance as therapeutic drug. So we have to get the substances that in these very active concentrations that are quite small will not affect the human cells. And here you can see an examples of some maybe most commonly used antibiotics. The first one, penicillin, historically the first one, and cephalosporin, with the basic structure that is seen here. Okay, we'll discuss them later on. So a few words about the history of antimicrobial therapy. So, of course, you know that within the whole history of the mankind, the doctors try to find some agents to effect to for treatment. But then the first really systemic attempt to find specific chemical substance to treat infectious disease was made by Paul Ehrlich, who searched for so-called magic bullet. Why this concept is called magic bullet? He dreamt of the substance that when we send it into the human body, it will destroy bacteria without giving any harm to the human body. That substance Paul Ehrlich had called magic bullet. And he did something in this direction. He coined the term chemotherapy and found a drug salvarsan that supposed to be effective against syphilis. Of course, it was quite toxic for the human body, but anyhow, it really was effective against Treponema pallidum, the causative agent of syphilis. Then, of course, we have to mention the name of Alexander Fleming, who in 1928 observed the anti-staphylococcal effect of uh, non-pathogenic fungi, penicillium, and this is a mold that you know already. And he was not the first one who had observed this effect, but he was the first one to recognize its potential for the fighting with the infections. But a long time takes, it takes a long time to develop the real drug. So, and only in early uh, 1940s, his idea came to fruition uh, when the method of mass production of penicillin was developed by him and his colleagues. So uh, I also want to mention the name of Gerhard Domag. who in 1935 discovered that just regular dye, red dye prontosil, with sulfonamide portion, and you can see the sulfonamide portion, uh, uh, and that is not really the natural product. So he demonstrated that it inhibits the growth of many gram-positive bacteria. And this discovery stimulated the development of a group of the substances that are called sulfonamides. And you can see the basic structure of sulfonamides that are very effective against a variety of pathogens. So by prevention of folic acid synthesis. So, and the development of antibiotics resumed with the work of the Salman Waxman, who was born in Ukraine and moved to USA in 1910. In, in the previous century, in 1940s, he discovered streptomycin and defined an antibiotic. And the streptomycin was also antibiotic, a result of natural activity of other microorganisms. And he defined an antibiotic as a chemical substance produced by microorganisms which has the capacity to inhibit the growth of bacteria and even destroy bacteria and other microbes or microorganisms in a dilute solutions. You see, nothing changed really. Streptomycin constituted a major breakthrough in the treatment of tuberculosis. And then also he worked and isolated chloramphenicol, neomycin, and chlortetracycline. And so he developed a large groups of drugs. So now we take a look at the basic property of the drug, and then we pay attention at some ways how can we categorize the antimicrobial drugs. The basic property of the uh, antimicrobial drugs is a so-called selective toxicity. That what Paul Ehrlich was dreamt of, because the selective toxicity is the selective inhibition of the growth of microorganisms without damage to the host. So it means that is the explanation of the thesis of magic bullet. And this is the most important concept underlying antimicrobial therapy. That's why the antiseptics are not really called, uh, cannot be really used uh, systemically, just on some surfaces, because they act not selectively against bacterial cells. Selective toxicity may be achieved by exploiting the differences between the structure and metabolic activity on the of the microorganisms on one side, whatever this is, bacteria, viruses, or fungi, and the structure and metabolic activity of the host human cell. So here we see that some examples of antiseptics that are non-selective, and these are chemical disinfecting agents that inhibit or kill most pathogenic microbes with sufficiently not so high toxicity for the host cell. And because this toxicity is controlled not so high, it can be applied directly on the skin, mucous membranes, or wounds. And antiseptics are comparatively less toxic with then the disinfectants. So that's a similar category of the drugs, of the agents without selective toxicity, but disinfectants are much more toxic. So they used usually to destroy pathogens on inanimate. You can see here already the examples of antiseptics, right? And with referring to their structure, for example, chlorhexidine, we can see it like here, and myramistine, the one that had been developed in our department with the former head of the department, uh, Professor Krivoshein. Today, antimicrobial or antibacterial drugs make a special group of chemotherapeutic agents used to treat diseases caused some microbes if antibacterial then caused bacteria. Very often they are called or refer to them as antibiotics. So, regarding of how these antibiotics are produced, they also can be categorized on the natural, synthetic, or semi-synthetic. So, what kind of subdivision? This is just a manner of their production. So, antimicrobial drugs, so antibiotics can be made directly from cultures of microorganisms, mainly fungi. Then will be called natural antibiotics. Then, by more by chemical modification of naturally occurred uh, substances produced by microorganisms. So we may get semi-synthetic antibiotics because with the help of chemical synthesis, we modify the nature, the structure, or using the natural substance and reading its structure, we may produce antibiotic by chemical synthesis. So, or artificially completely, but still it will be antibiotic, but we would call it synthetic antibiotic because as a template or as an ideal structure, the natural uh, substance is used. So that's one way of categorizing of antibiotics. Then what is important in microbiology? Antimicrobial drugs can be also categorized depending on the spectrum of the activity, here number one, character of activity, so how do they work, and mechanism of action. These three is mandatory, so now we are going to discuss them. Spectrum of antibacterial activity. We may have broad spectrum and narrow spectrum antibiotics. So, broad spectrum drugs are active against several types of the microorganisms. And here you can read the examples of these broad spectrum antibiotics. And here you can see an example of narrow spectrum antibiotics, those that are active against one or very few types of microorganisms. So, for example, gram-positive organisms only, and this is against different. Now, let's take a look at what do we call different types of microbes, of bacteria. See here uh, we demonstrate some types of bacteria. Why we divide bacteria into these large categories? For example, that's understandable. Gram-negative bacteria and gram-positive bacteria. The first gram-negative bacteria, they have very thin cell wall with a complex structure, but then the peptidoglycan layer is very thin, right? And gram-positive bacteria with simpler structure, but the peptidoglycan layer is very thick. These are two basic so-called types of bacteria. But then we also may have bacteria, not regular bacteria, that may differ in the cell wall structure, but also that may differ in their lifestyle. For example, chlamydia and rickettsia, these are bacteria that live inside the human cells regarding the human pathogens. So it means they are obligate intracellular parasites, but then they are still bacteria with a structure similar with that of true bacteria. So then we are going to affect them when they are already inside the human cell. So they make different type of bacteria. But then we're supposed to have wide broad spectrum antibiotics that are active, for example, like tetracycline, against gram-positive, part of gram-negative bacteria, chlamydia, rickettsia, and so on. Even penicillin that you are familiar with, they are effective against part of gram-negative bacteria, gram-positive, and even partially against chlamydia. So then, but also we may have narrow spectrum antibiotics that are solved to treat particular type of bacteria. For example, take a look, isoniazid. It is very effective against mycobacteria, and you see here that regular antibiotics, they are not effective against mycobacteria. Why it is so? Because mycobacteria have a very special structure of the cell wall, and they are different in their chemical composition. So not any type of antibiotic may affect mycobacteria. So sometimes we may use narrow spectrum antibiotics that affect this particular type of the organism. So the second way, common way of categorizing antibiotics is to categorize them according to the character of activity. Do you remember in the definition what is antibiotic? We give the character of activity. So it may kill bacteria or inhibit their growth. That is the clue for understanding of the character of activity. We may have some drugs that kill bacteria, you can see it here, and you can see here the examples of so-called bactericidal drugs, those that kill bacteria. And also we may have drugs that in active concentrations that are used in humans will not kill bacteria but arrest their growth. And you can see here the list and examples of antibiotics that cannot really kill bacteria in uh, safe concentrations, safe for humans, but just inhibit their growth. So, bactericidal drugs kill bacteria and they destroy bacteria in our body. So, when we prescribe them, we account on that activity that they will kill bacteria. By then, if we use bacteriostatic drugs, so normally bacteria divide like this, multiply by binary fission, but then what we account at when we use bacteriostatic drugs? We stop their multiplication. But then bacteria are not killed, they stay in the body. So what we account at when we want them to be eliminated? So we think that host defense mechanisms, so phagocytosis and some other cells and substances will be required to kill these bacteria, and we just stop their growth. So, but then we have to take into consideration that if we use this type of drugs, when we stop or withdraw uh, the drug, then bacteria may grow again if host defense mechanisms not effectively eliminated from the body. So, and finally, that is the very important for us uh, way over the bacteria antibacterial drugs classification is according to the mode of action, because we are studying bacteria or studied already bacteria, so we have to understand on what exactly structures and metabolic pathways antibacterial drugs act, so to inhibit the growth of bacteria or to kill them. And here you can see a table with the most common modes of actions over the uh, mm, antibiotics. Then, for example, so the best target for the pharmaceutic uh, uh, market, that is the to produce antibacterial drug that affect cell wall synthesis. So this is a cell wall of bacteria. So, and we have a lot of examples, and they may affect the cell wall synthesis in several ways. But then the target is same, the cell wall synthesis. So then, if we would like to, if, for example, the teacher asks, what is the basis for selectivity of this drug? So we have to think of the differences in between the bacterial cell and the human cell. Why until now the drugs that affect cell wall synthesis are considered least toxic? That is the very high selectivity of the toxic activity. That's because the bacterial cell has the cell wall made up of the peptidoglycan. But the human cells, they don't produce this uh, structure. They don't have cell wall at all in the human cells. It means the human cells don't even have any target for activity of the drug that affects cell wall synthesis. This makes these drugs very uh, usable, most usable maybe, because they are highly selective against bacteria and won't affect the human mm, cells. Then the second very good target is a protein synthesis. And here we may have the drugs that affect uh, the organelle that where the protein synthesis occur, 50S ribosome. And there's a lot of, and very nice, very large groups of macrolides, chloramphenicol and others. Then we also have substances, they affect the 30S subunit, and these are aminoglycosides, tetracyclines, very large groups of antimicrobial, antibacterial drugs. Then we can see that the uh, mm, drugs may affect the layer beneath the cell wall structure, that is cell membrane. These are those drugs that disrupt somehow and affect somehow the cell membrane. So, but then you can see or you can compare how many examples we have here and there, right? So, but then you can see that very few examples we have here for cell membrane disruptors. Why it is so? I think you have an answer. You also understand the answer for this question because the cell membrane of the bacteria is not that different from the cell membrane of the human cell. So that's why the cell membrane disruptors to some extent and or to high extent will affect also the cell membrane of the human cell. So that's why we don't have that much drugs that are used against bacteria because so many other targets are accessible. So, but then we may find, if you remember that we mentioned when we discuss a bit antifungal drugs, but we have quite a lot of antifungal drugs that affect the cell membrane. So, because fewer targets can be found in the in for antifungal drugs, because fungi, including pathogenic fungi, are eukaryotic organisms, but humans are also eukaryotic. So, uh, less differences are observed in the structure and metabolic activity of the human cells and fungal cells. Uh, by the way, come back to the protein synthesis inhibitors. So they are also um, selective, and they are highly selective. What is the basis for their selectivity? So do you remember in the first lecture in our department, and of course in biology department, you heard that the ribosomes of bacteria um, are different from that of the human cells, right? So here we have 50S and 30S, altogether 70. And in human cells, we have different size of the ribosomal subunit. So that's why those drugs that we use, they have priority to affect bacterial ribosomes or subunits of the ribosomes to compare with the human cell ribosomes. That is the basis for selective toxicity. Differences in structure of ribosomes responsible for protein synthesis. So for cell membrane, we have discussed the basis for selective toxicity is not that good, so fewer drugs can be developed. Then the nucleic acid synthesis. And here we can have the drugs that affect the DNA synthesis and the drugs that affect RNA synthesis. And also one of the not very common but targets for antibacterial treatment is the mainly here we can see the structural uh, differences, but this is demonstrate or development of this group of drugs demonstrate the using differences in the metabolic pathways of bacterial cells and human cells to inhibit the metabolic pathways of bacteria that don't exist in the human cells. So later on we'll discuss the activity of the sulfonamides and trimethoprim that affect the folate synthesis. So here we can see the basic um, targets for these drugs, and we discuss also the selectivity of these drugs. Now let's take a look at the uh, basic groups of the drugs. So of course, first one and the one that we discuss more accurately is the penicillins, that is the group of antibiotics 10 taken from the penicillium fungus cultures, or they can made in the laboratory, that will be synthetic penicillins or semi-synthetic penicillins. Then another group is quite similar, quite close to the penicillins, is cephalosporins. Is an antibiotics originally derived from the fungus Cephalosporium acremonium. Cephalosporins are similar in chemical structure to penicillins. Penicillins and cephalosporins are bactericidal, and it means that they kill bacteria and act by inhibiting the particular enzyme that is called transpeptidase. What is the function of this enzyme? On this enzyme, if you remember the structure of the cell wall uh, of bacteria and the structure of the peptidoglycan, so you're supposed to remember that the basic bone backbones of glycans is cross-linked with the uh, polypeptides. This transpeptidation or cross-linkage occurs by transpeptidases. This drug kill bacteria only when they are growing, because exactly in this moment the cell wall supposed to be in process of production, and transpeptidases are working actively on it. So in non-growing cells, the penicillins don't perform the cross-linkage, so they are not really effective in non-growing cells. Here you can see the particular targets in the peptidoglycan for the penicillins. So because penicillin binds to a variety of the receptors in the bacterial cell wall and cell membranes called penicillin binding proteins. Some of penicillin binding proteins are so-called transpeptidases that catalyze this linkage. So changes in penicillin binding proteins may contribute to an organism resistance to penicillin. So if it won't bind with it, so it will be difficult to affect. So uh, some additional factors may contribute to the activity of the penicillin. So autolytic enzymes called murine hydrolases are also activated in penicillin-treated bacterial cells. So there's an additional effect of the penicillin therapy, because these enzymes degrade the peptidoglycan. So activation of autolytic enzymes in penicillin-treated cultures. This is additional way how penicillin works. Then what happens with the cell? When the cell wall synthesis is inhibited, you can see staphylococci, and that's how they look under the uh, high magnification microscope before treatment, and that's how they look with the same magnification after the treatment. You can see that the affecting this the cross may make you know, make the cell more permeable for uh, water solution, and the cell cannot stand the osmotic pressure. Water comes inside the cell, so the cell lose integrity, and then it start to be enlarged, and due to water influx, and then uh, they rupture because of the high osmotic pressure. Here you can see the basic structure, and you can see the similarity in basic structure of penicillin and cephalosporin. This power portion is called beta-lactam. So that's why those drugs are also called beta-lactam drugs. So, and this is active portion of the antibiotics. And intact structure is essential for antibacterial activity of these antibiotics. But then it appears that some microorganisms may produce enzyme that is called beta lactamase. What is the result? If beta lactam ring, here the blue one, will be broken, then it make penicillin or cephalosporin non-active. By the way, we studied, or you've heard the lecture on genetics already, and I'd like to remind you that we may have some plasmids that keep information about the production of the beta lactamases, these enzymes. So, and the ability to produce beta lactamases can be transmitted by the bacterial plasmid from one culture to another culture. So, here we can see several examples of natural semi-synthetic penicillins and also cephalosporins. Then why we have so many analogs and so many generations? By now we have 3, 4 generations of the penicillins and cephalosporins. So because the uh, effectiveness of the drugs can be increased by chemical changes in the side chains. You can see it here, right? And you can see the chemical changes in the uh, side chains of the penicillins and cephalosporins. Why we need those changes? First, they may decrease the excess of the beta lactamase, bacterial enzyme lactamase, to the beta lactam ring. So the bond can be stereometrically protected by the modification of the side chains. They start to be larger, uh, and with the addition of aromatic rings or long uh, carbon backbones, so methyl and ethyl groups. So it may make the uh, beta lactam less accessible to beta lactamases. And of course, apart from this, the uh, pharmacodynamic can be increased with some synthetic modifications of the drugs, and the ability to penetrate, for example, in the cerebrospinal fluid and so on, right? So that's why we have so many generations of them. Then the second type of drug or group of drugs that inhibit of protein synthesis. Uh, so, and they do, usually the drugs do it selectively due to the differences in between bacterial and human ribosomal proteins, so with differences in RNAs and associated enzymes. So enzymes associated with the process of protein synthesis. So then inhibitors of the protein synthesis are commonly divided into two categories. 50S subunit inhibitors and 30S subunit inhibitors. 30S inhibitors, an example of it, for example, aminoglycosides. These are bactericidal drugs like streptomycin, amikacin, and the others. And they are very effective against gram-negative rods and certain other bacteria. So, but any drug has its own limitation. For example, uh, those drugs may have a toxic effect on kidney and on the auditory and vestibular portion of the eighth cranial nerve. They can be poorly absorbed from the GIT, so that's why usually are not given orally but systemically through injections. So then they may not be very effective against anaerobic bacteria, and they should not be used for um, uh, treat bacterial meningitis because they poor penetrate the cerebrospinal fluid. So, how do they work? They inhibit the initiation complex composed of the uh, 30S uh, and 50S subunit and mRNA. No peptide bonds will be formed and no polysomes are made. So then, another way is misreading of the triplet codons over the on the ribosomes, and as a result, the wrong amino acid can be incorporated into the protein molecule. As a result, membrane damage occurs and the bacterium may be killed. So, but then, some we said that quite often there's no absolutely harmless drugs. So, of course, they mainly the majority of them can cause some side effects. For example, aminoglycosides are very good drugs, but they may cause very severe allergic reaction that you see here. The one more group that's a very common, old, and very useful group is the tetracyclines with very broad spectrum activity. The character is that they are bacteriostatic against variety of gram-positive and gram-negative bacteria. They also may affect mycoplasma that naturally don't have cell walls, so penicillins and cephalosporins are useless for them. And they also um, effective against chlamydia and rickettsia. They inhibit the protein synthesis by binding to 30S ribosomal subunit and blocking transport RNA. In generally, tetracyclines has lower or have lower toxicity. And the selective action of tetracyclines on bacteria is based on the greatly increased uptake into bacterial cell to compare with the human cell. So they inhibit, uh, okay, we did it already. So, but still, even that very nice, very good drug and very effective, have some limitations and may produce some side effects. For example, because they are broad spectrum, they may cause the suppression of normal flora bacteria during the treatment in the intestinal tract, which can lead to diarrhea and overgrowth by drug-resistant parasitic bacteria or fungi. See, as a result, this is so-called black hairy tongue. So that is overgrowth of fungi. So, and then they may cause brown staining of the teeth of fetuses and young children as a result of deposition of tetracycline in developing teeth. So that's why it is not recommended for children younger than 12 years old. So then we have drugs that affect 50S subunit, and one of the examples is chloramphenicol. This is antibacterial and it is effective against rickets. They're also bacteriostatic drug, and also they can act against broad range of gram-positive or gram-negative bacteria, including anaerobes. So it has bactericidal activity against very important pathogens, the causative agents of meningitis, because it can penetrate into cerebrospinal fluid. So this drug inhibits the protein synthesis by binding to 50S ribosomal subunit and blocking the action of peptidyl transferase. This prevents the synthesis of new peptide bonds, so the synthesis of the protein is blocked, arrested completely. So, but then this is a good drug, but we have some limitations. It may produce dose-dependent bone marrow toxicity. So the drug inhibits protein synthesis in mitochondria, also human cells, because there is a large similarity between the ribosomal mitochondrial ribosome that have 50S ribosome that smaller than the other one, and uh, among the serious side effects of chloramphenicol we may have as a result the blood disorders, aplastic anemia, and so on. But then sometimes we may use it as a drug of choice. For example, in some diseases like typhoid fever, still chloramphenicol will be a drug of choice. Then a very large group that affect 50S ribosome is called macrolides. See, the macrolides, large aromatic rings are incorporated into the structure. So they are bacteriostatic drug with very broad spectrum of activity. So these drugs inhibit the protein synthesis by binding to 50S ribosomal subunit, blocking the polypeptide chain elongation. So macrolides are effective against a lot of pulmonary pathogens like Mycoplasma, Legionella, Chlamydia, and some of them are intracellular parasites like Legionella and Chlamydia. And they are very effective against gram-positive bacteria. The first discovered macrolide is erythromycin, is one of the least toxic substance of commonly used antibiotics. But unfortunately, as usually, resistance can be developed. Then we discuss several uh, uh, issues regarding uh, resistance later on. So then, inhibition of nucleic acid synthesis can be achieved by around three mechanisms. Inhibiting DNA synthesis directly, RNA synthesis directly, or inhibiting the development of the precursors or para-aminobenzoic acid synthesis. And these are phosphoramide and trimethoprim. My dears, you can see here that sulfonamides are structural. See, there's a structure of sulfonamide, and then there's it is analog of the uh, para-aminobenzoic acid that is essential in the microbial metabolism that yields folic acid. So when sulfonamide is incorporated instead of uh, PABA, then the synthesis of folic acid is prevented. Then the other one that acts in a bit similar way, This is a trimethoprim on similar pathway. Inhibits the enzyme dehydrofolate reductase. That activity of this enzyme is to prevent the conversion of the dehydrofolate CDTH into tetrahydrofolate that uh, is important in the transformation of the folic acid. So, and we have a lot of drugs under in this group, and quite often they act synergetically. Sulfonamides with trimethoprim, several generations of them, uh, mm, and the uh, basis for selective toxicity of the drugs is that the folic acid is not produced in mammalian cells and taken as vitamin. So, uh, they are very effective against broad range bacteria, including chlamydia. But then also the uh, resistance can be developed. So, some drugs like rifampin, it affects the RNA synthesis. Uh, It is a semi-synthetic drug that binds to DNA-dependent RNA polymerase and inhibits the initiation of RNA synthesis. So, uh, as usually, it has some side effects. For example, for rifampin, it is typical red man syndrome, which occurs when colored metabolic products of the drug accumulate in the body, eliminated through sweat glands. It can also cause liver damage when high doses are used. So then some drugs may affect DNA synthesis like quinolones, but I would like to tell you that quinolones are not antibiotics. They are antibacterial drugs, but they are completely synthetic and they inhibit the bacterial DNA topoisomerase, which is required to supercoil the strand of bacterial DNA. And these are the examples. The first one was nalidixic acid, uh, and then for chemical modification, the other members of this group had been developed. But unfortunately, the resistant also can be developed. So there's a one more picture that explain the mode of action of the drugs. To summarize it, you can see here cell wall synthesis, uh, DNA, RNA synthesis, folic acid metabolic activity, protein synthesis, and the uh, cell membrane disruptors are not shown on this picture. So we go ahead. Mechanisms of drug resistance. So here you can see, and yes, I would like to tell you that drug resistance of bacteria can be considered on two levels. The first level is genetic level. So it means if something happens in the bacterial body, in the bacterial cell, of course, first some changes should occur in the genetic material. And then when these genetic changes will be physically expressed, we will see some biochemical mechanisms that may make bacteria less sensitive to the drug. So let's start with the genetic mechanisms. Genetic basis of bacterial drug resistance. First, and most important one, the resistance can be plasmid mediated. So it is very important for several reasons, because the exchange with the plasmids may occur between related different species of bacteria. For example, this process is very common in gram-negative rods. For it, so it means, to simplify it, The normal flora Escherichia may uh, um, share the plasmid with pathogenic salmonella if they meet together, right? So that's the uh, transfer of the plasmid from one species to another one. Then the second reason is uh, the ability to stand several types of drugs may be fixed in one single plasmid. Then the transfer of this plasmid will cause multiple drug resistance from one single event of the plasmid of the conjugation. And then the third reason is that plasmids has a very high rate of transfer from one cell to another cell. So this process occurs very actively, naturally in the bacterial populations. So very frequently the antibiotic resistance can be transmitted. Then the second important genetic basis of bacterial drug resistance is the spontaneous mutations in gene that codes for the target of the drug. So, for example, uh, if the mutation occurs in the gene that codes for DNA gyrase, the enzyme that's supposed to supercoil bacterial DNA, then this enzyme cannot be affected by quinolone. Then bacteria will produce functional supercoiled DNA molecule. So chromosomal resistance is less of clinical problem, but in some types of bacteria, for example, in mycobacteria, mainly the resistance is developed by mutations in chromosomal genes. So that's why it is also very important. And the third basic mechanism is transposon-mediated resistance that is slightly different but common with that produced by the plasmid. Then this was the first level. Then we take a look at what these changes in genetic material may lead to. So take a look at this picture. We have an examples how the changes in the genotype will be expressed in a phenotype. So the plasmid mediated resistance can be can lead to, for example, production of beta lactamase. So the information about beta lactamase can usually stored in the plasmid. With the transfer of this plasmid, the bacterium start to be resistant to such drugs as penicillin and cephalosporin. So then, example mutations may change the sensitivity to antibiotics. For example, mutations in certain bacterial genes may lead to synthesis of modified drugs. For example, this is typical for aminoglycosides, erythromycins, and as we said, quinolones, where the peptidases, if they used as targets for the drug, if they will be uh, if mutations occurs in the gene that code for these enzymes, then these enzymes will not be affected. That is called modification of the drug target. So then mutations also may cause caused in the gene that encode porin proteins may affect the ability of the drug to penetrate into the bacterial cell. And if they reduce the amount of drug entering the bacteria, of course, the effect of the drug will be reduced. And that's typical for penicillins, aminoglycosides, and others. And this is called reduction of permeability of the drug. Here on this picture, you can see some examples, right? Inactivation of drug by enzyme. Usually, this is produced by plasmid-mediated resistance. Then, the activation of drug efflux pump or alteration of drug target quite often occurs with the involvement of mutations. And inhibition of drug uptake also. Then these are some biochemical mechanisms that is the result of particular changes in the genotype and the expression of these genes. Okay, my dears, we go ahead with the antibiotic sensitivity testing. The thing is that that is a very important laboratory manipulation because in ideal, so we have to check every single strain of bacteria that we isolate from the patient. We have to check to what antibiotics this particular strain is sensitive or resistant, because we need to choose the groups or types of antibiotic that we're supposed to use in treatment. Uh, because microorganisms may develop resistance. Usually laboratory tests are needed to show which antibiotic kills the microorganism, and for this we usually isolate the microorganism from the patient first. So the next test supposed to be done using pure culture, my dears. This regular disk diffusion or Kibbe-Bauer method and serial dilution method. Let me check. Okay, so this diffusion method, what we do, uh, we first perform a culture of bacteria on the petri dish on the agar medium, and we perform a so-called carpet culture because we place a drop of bacteria, broth culture of bacteria, and spread uniformly over the plate. So, because we would like to have a carpet of bacteria in here, right? Then we place the, see here, the discs that are soaked or saturated in the antimicrobial drug. And then we put a plate into the incubator for bacterial growth because bacteria need to grow. Once they grow, so they spread within the plate, but then if they cannot grow in the presence of the antibiotic that diffuses during the incubation from the disc into the agar medium, so then they stop growing. And then we see the clear zone of no growth of bacteria around the disc. This zone is called zone of inhibition. Bacterial growth is inhibited. So, and you can see here. So, the different zones of inhibition are formed there. But the thing is that we cannot compare directly and make conclusion. If the zone, we measure the zones. If this zone is larger than that one, so this antibiotic is more effective. No, it is not correct. So we have to measure the zone and compare it, the diameter, with the standard table where the same bacteria is tested that doesn't have any resistance against drugs, against different type of antimicrobial drugs. And it appears that if the diameter in the experiment is equal or larger than that in the standard table, then the bacteria are very sensitive to antibiotics. But if there is no zone of inhibition, or this zone is, the diameter is smaller than that in the standard table, then it means that our patient bacteria develop resistance against this drug. Okay, and the next method is a serial dilution method. So we isolate microorganisms from the patient and perform serial dilutions of this and perform serial dilution of the drug. So usually twofold dilutions. And then we add this patient bacteria in the same concentration and same numbers into each tube. Okay, we have it in tubes here. That's why maybe it's more convenient for you. See here, these are the dilutions, right? So this is uh, so these are the dilutions. Okay, so then we add bacteria, and you can see whether and after incubation we see whether the tubes are transparent or opaque, turbid. Because if the tube liquid in the tube is turbid, it means bacteria are growing there, multiplying there. Then if the liquid is clear, so it means that the bacteria are not growing there. So then we consider that we have bacteria in the first, second, and third dilutions, right? So let's say this is 2 milligram per ml concentration. But then in concentration of the drug, from here. So 4 milligram. So there is no growth of bacteria. That can be performed also in the plate, right? So we see here, we can see the dilutions of the drugs. And then we consider penicillin drugs, bacteria growing, 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 growing. No, here they are stop growing. So we don't see bacteria in this dilution. So here, cephalexin, bacteria grow here, but then they won't grow in other dilutions, starting from 1 is to 16, right? Then we can find minimal inhibitory concentration. This is a minimal concentration that stop the growth of bacteria. For penicillin, it is 1 to 1. For uh, cephalexin, it is 1 is to 16. For ampicillin, it is 1 is to 16. For methicillin, is 1 is to 8, right? So here we have increased dilutions, and here we can see the growth of bacteria. So then, if we don't have the growth of bacteria, then the question is, how do we know whether bacteria are alive there or not? If they are still alive, so it is bacteriostatic drug. If they are killed, they are bactericidal drug. So then to identify whether this drug is bactericidal or bacteriostatic, we have to take a sample from the tubes used for minimal inhibitory concentration assay and spread it over the surface of the drug-free some agar medium, right? So then what we see, that if we place this sample from the tube with minimal inhibitory concentration, so the first one where we don't see the growth of bacteria here, it is 4 milligram per ml. If we then culture put this plate into the incubator, we see that bacteria is still growing there. So it means in its minimal concentration, this drug inhibit the growth of bacteria but doesn't kill them. If we use the next tube with a lower concentration of drug, higher dilution, and again place it on the agar plate and incubate it, we see that still we have the growth. So in the dilutions, 1 is to 4, 1 is to 8, and 1 is to 16, we don't see, we see the growth of bacteria. So all those concentrations will not allow us to kill bacteria. Then finally, when we find the tube, when we take a sample, there will be no bacteria in the next one. So then we find minimal bactericidal concentration. This will be minimal concentration that kills bacteria. If the minimal bactericidal concentration in our case is 1 is to 32, so then, and it is much higher than the minimal inhibitory concentration, then it is bacteriostatic drug. And if it is lower, then it is the bactericidal drug. With this, we have to finish our lecture, and nobody really attended the lecture. I just have some hopes that you take a look at this lecture later on when you will be free, maybe in the evening time. So, goodbye, my dears.